Hi, everyone. I'm Brad Power, and this is the Cancer Patient Lab. And we're honored today to have with us Joanne Weidhaas. She's at UCLA, and she's got a diagnostic company or service that she's going to be describing to us. It's of interest in its own right, but it's also of interest because of the novel ways that she's bringing it to market and getting patients early access to it through things like a clinical trial. So this is going to be a fascinating conversation, both about the technology, but also about how you bring a technology to market. Just before we get started, a few of our standard housekeeping things. This is for information purposes only. It's not medical advice. We try to arm patients with information they can bring to their medical team. And second, this is going to be made public. Everything you say can and will be used. And so if you're concerned about having your image or your name put out there, you can hide your name, hide your image, and you can not say anything. And finally, we are a 501c3 nonprofit, and we depend on the kindness of strangers, as I think they said from a streetcar named Desire. And so if you would be interested in donating, it's easy to do through the Cancer Patient Lab website where there's a donate button. With that, I'll turn it over to Joanne.
Great. Thank you so much. Can you hear me all right?
Hear you.
Good, great. I'm going to tell you just about a journey that I've taken, which I don't know if it's probably atypical because I'm stubborn, but I'll tell you kind of where we ended up, where I really have a company, a molecular diagnostics company that I co-founded called MSM, but also a nonprofit called Me Our Kind, and I'll tell you about how we got there. So just my background, I'm a physician scientist. I'm actually a radiation oncologist, who, as I said, my view was that I'm going to do good science and become really an expert in cancer, which was always my interest. And with that, if I find something, that will be a dream and it will make it to patients. And that's all that matters. I had no idea that how complicated that was, in which I'll tell you a little bit about. My co-founder of MiraDX, his name is Frank Slack. He actually, strangely, did his PhD across the hall from me at Tufts. He's a PhD developmental biologist, and he actually discovered the first human microRNA, which is a new understanding of genetics, which the Nobel Prize was just awarded to his boss, Gary Revkin. Frank discovered LET7 during his postdoc work with Gary, but of course the boss always gets the Nobel Prize, but that's just how it goes. So, Frank and I had known each other and were friends. And then when I went to Yale, Frank was there. We began working together, trying to understand how we could apply microRNAs in cancer. And I'm an oncologist, as I mentioned, and Frank is incredibly knowledgeable on the science side. So I'm just going to give you one slide about science, just so you know kind of what we're talking about. And then I'll go back more to the challenges. So what we've known about our DNA, we have the same DNA in every cell in our body, and the parts in your eye tell your eye how to be an eye, and your liver DNA tells your liver how to be liver cells. But what we've known for a very long time is that DNA makes messenger RNA, and that makes protein. And that was really the holy grail and what everyone thought. But over the last 2.5 decades, it was found that actually we use a lot of DNA that doesn't turn into messenger RNA. And they call it non-coding RNA because non-coding DNA, actually, that's a typo, because it doesn't turn into protein. But it turns out that's really important. And this DNA that we've been working with for the last 70 years, that part of the DNA, it's only 2% of all of our DNA. And any tumor biomarker that you can name, so HER2, EGFR, BRCA, it's in this little slice of our DNA. And that's where all the studies were taking place. All the drugs are targeting these proteins. When really it turns out that the non-coding DNA, these non-coding RNAs they make, microRNAs were the first examples, they're really the boss of the messenger RNAs. So they really control things. These are really just the tools, the messenger RNAs. So, but this is new. The first microRNA or non-coding RNA was found in 1996, in humans shown in 1999 by Frank Slack, as I mentioned, and Gary Ruvkin. And we now know that that makes up about 80% of our DNA, 78% of our DNA makes these biomarkers. And there's just a wealth of data in them because they're really kind of in charge in the cell. So, with this information, there's so much more that we can learn from a patient themselves. That this was our concept, that at least at this point, we realize this is our concept, that we could actually look in the DNA in a normal person, not just in a tumor cell, but in a person. It's easy to test somebody's DNA because you can do it in a cheek swab. And then using this important parts of the DNA, we could actually find answers that we need in cancer. And so I'll tell you a little bit more about what we've done with that. So, and part of that is backing up. So the way that cancer therapies have been developed, and this is a little different with immune therapy, but historically, we look at a cancer type, lung cancer, ovarian cancer, breast cancer, we'd say, let's look at the tumor and find a difference that we can target with a drug, estrogen receptors, EGFR mutations, HER2 mutations, PSA actually for prostate, even targeting that. And then let's develop drugs, and it will be specifically for a patient with that tumor type with those biomarkers. So it's a subset of people. They can be very successful, some of these targeted therapies, but it doesn't really tell us about the patients themselves and how they're going to respond. So, with our concept and what we have been working towards is that you take people and, okay, they have cancer. We look at their DNA and we find out how they would respond if we gave them radiation, if we gave them immune therapy, if we gave them chemotherapy, as a whole group, as people. So this all sounded like a great concept. And honestly, to be honest, I had never ever planned to start a company. I'd never even taken an economics class. I really believe this. If I find anything important for my patients, someone's going to take it and do great things and make sure it gets to the patient. My heart was always with, let's solve the pain points patients have. And to me, I did my residency at Sloan Kettering, and what I saw was, one, no one expected they were going to get cancer. So we clearly didn't understand why people got cancer. I have so many healthy cancer patients. And two, we'll treat people the same way and they will have incredibly different responses. So there'll be people that do wonderfully and are cured and have no side effects. There'll be people that do poorly and don't respond. And there'll even be people that respond but have terrible side effects. And they're all treated the same. But there has to be answers. And I just really believed they were in the patient and we weren't seeing them because we're looking at the tumor. So, we found these examples with these new biomarkers, and I could see we have to get this information to patients. This is a real change in how we even think about identifying the best way to treat people. So with that, we started a company. Frank and I started this company in 2008, which is forever ago now, very naively. And I would say my husband is a lawyer and does private equity. So he, knew people that could raise VC money and all that. And we started kind of quickly realize, so that's a normal path. You have a concept, you spin it out of a university, which we did to start a company. You find a management team who goes and raises venture capital money because they have to build it up for someone to buy it. Like, that's generally the path. I felt very uncomfortable with that. Frank and I really, our goal was to get the information to people to help them. And it was pretty clear that the job of the venture capitalist was to make money. That is their job in general. And that wasn't what was motivating us. It was really, we wanted to get the information to people. So we ended up not taking venture capital money. We raised friends and family money. We built this lab in New Haven, got it CLIA certified, and still I thought, our first marker actually predicts ovarian cancer risk. I thought this would be great. We're going to get this to patients. This will be really meaningful. But we needed business people. I wasn't a business person, and maybe it's hard to get business people in New Haven, Connecticut, where the company was outside of Yale. But they went the standard path. They said, okay, we're going to get a lot of orders in. We'll submit them to insurance. We'll build up so somebody can buy us. And it was very expensive to do it that way. And we were just completely naive, and they were naive, and we ran into there was a huge competitor, which was Myriad at the time. Of course, insurance wasn't going to reimburse us, and we kind of hit a wall in late 2011. And I thought, God, why is this so hard? I know we have something that could actually help people, and I can't get it to them. It was so baffling to me. I had never experienced that, not being a business person. So I actually took a leave from Yale to go to the Stanford has a 1-year program. It's called X now, I think. But to Stanford Business School, picked up the family, moved to California, just to figure out why it was so difficult. And I learned a lot of really important things in that year. The main thing was that even though our first test predicting ovarian cancer, it was really for the patients. Medicine's complicated. You cannot have something that's only for patients, even though that's where my heart lies. It can be for patients, but you need a doctor to order it. So the doctor has to see a benefit and know how to use it. And then neither of them pay for it. Insurance pays for it. So it's very hard to have an aligned system. And for cancer prevention, which was our first test, that is just inherently misaligned. Insurance doesn't want to pay for a test that tells a patient they're at risk of cancer, because then they're probably never going to leave. And that's a bad patient to have stuck on their insurance plan. So, with that, I actually, in 2013, spun out the nonprofit, which I'll tell you a little bit about, Mirakind. And I have to say, at the time, I realized that maybe I probably should have just started a nonprofit, because it really wasn't about money for me. It was about trying to help as many people as possible. But through Mirakind, that really enabled us to just focus on patient education and access for that first test about cancer prevention for women. I also realized that I would never be compatible with a VC funding model, which is diagnostic companies, what they do, what the, formula has been, is they raise a lot of money because they hire a lot of salespeople, 100 salespeople. They spend $100 million to build business, finally get insurance to pay, and then they go public or they sell. And I thought, God, for money like that, think of all the tests we could give away. So I'd rather take the slow path and try to get people involved, find ways for them to get it with something that's useful for the patient. We can find something that doctors want to, that ultimately could even save insurance money. So, I left business school with a much better understanding of the complexities of our medical system. And it's not for want. People have great ideas and great findings, but it is so hard to cross that. They call it the valley of death, which is appropriate. It's just so hard to get out there. It requires a ton of money. So you're kind of selling your soul to venture capitalists, which, do get things to patients. So I'm not trying to say bad things, but so very challenging. So, with that, I had the nonprofit, I had this understanding that if I was going to have tests, they needed to align the whole system, the ecosystem. I came to UCLA, left Yale, came to UCLA as head of translational research, and made very clear to them, this is what we have. I have this new class of biomarkers. I want to apply it for patients. I think it'd be really useful. I'm a radiation oncologist. In radiation, we have patients with very different responses and very different toxicity from treatment. That's a great use of these markers. Moved the lab from New Haven to Los Angeles, got it set up here. Then for the next 4 years, we just lived on government funding, SBIR grants, which are actually a nice way to survive. So we survived, had wonderful collaborative studies with UCLA, which I still do. Our first product actually is in prostate cancer, and it identifies patients who will have different toxicity to different types of radiation treatment versus not. So you can choose the safest path, which is a great alignment of course, the patient wants to know, get the non-toxic treatment. The doctors actually want that too. And actually insurers want that too, because paying for a treatment and then paying for toxicity is more expensive. So it was our first aligned ecosystem aligned test. We took a detour in 2020 because of COVID and actually ended up doing an enormous amount of COVID testing and only pivoted to do that to help. And it ended up being something that was all encompassing for a couple of years. Meanwhile, our first test, Prostox, went through a nice prospective validation study. This fully funded the company, which is wonderful. So we're secure. And then since 2022, we have done a few things, and this is where it ties it all together. And then I'd love to look at some of the questions here. We, and I'm like, I don't want salespeople. I only want people asking for offering the test to their patients because they think it's useful. And I want to know if they think it's useful. I think a patient will think it's useful. So we started an early access program, which I don't think anyone does unless insurance makes them, but I'm like, this is a great way to work with smart people, to see smart physicians, to see how they like using this and have them talk to their patients about it. So we have about 15 centers, the leading centers actually, radiation oncology clinics that are able to offer this test to their patient, Prostox, and get back the information, share that information, and do joint decision-making with the patient, which I think is ideal. UCLA orders this test on every patient coming for prostate cancer, but it's also used at the Mayo Clinic and UCSF and just all over the country at this point. But it was great, and we didn't bill insurance, so it was great for everybody, great for the doctors, great for the patients, most importantly. And at this point, we're just building our data and not fighting with insurance, so no one's being stuck with anything. However, about a year ago, we had a lot of patients. Prostate cancer is interesting because patients are diagnosed by the surgeon, and they don't all get to go to the radiation oncologist. Half of people get surgery, half of people get radiation. And many patients were reaching out saying, I really want to get this information. I haven't seen a radiation oncologist. So that's where we actually really were able to pull in the nonprofit Mirror Kind, which is all about this enabling access, because we have a study through, and I'm like, let's figure out how to make this work. We have a study through Mirror Kind where a patient can come to Mirror Kind, join a study. There's a cost to it, but pay for their tests and by joining a registry. So it allows access. And then I'll end with just a brief. So MirrorDX, I told you a little bit about it. If you just looked at MirrorDX, I'd say it's, Frank and I founded this company to really advance and improve human health in this new class of biomarkers. But, we're really partnered with MirrorKind, which has a very clear mission, which is to really educate people about this new understanding of DNA, to advance research. There's a lot of research studies in many cancer types that goes on through MirrorKind, but actually really to enable access so patients actually can get meaningful information. And so it's just this two-pronged model. And to me, it was never planned this way. The goal was to make sure we can get important things to patients and do it above board so we don't get in trouble, responsibly, of course. And so it's been a fantastic partnership. And of course, we have great legal counsel to make sure we're following all the rules to do it. But I'm feeling like we're getting there, which is incredibly rewarding. It's been a long, long journey. And that's it.
Thank you, Joanne. We have a number of questions in the chat. We also use the raise hand feature, and I'll help moderate.
Fantastic. I don't know if you want to read the questions in the chat?
Yeah, I'll feed you the questions so you don't have to be reading the chat and everything. Just one from me. So I recall when you and I spoke, you also had a technology for predicting toxic reactions to immune checkpoint inhibitors. Can you
We
also
do.
speak to that one? Because I think that's a pretty big area, but it's and wider than prostate cancer
For
patients.
sure.
Yeah.
It's interesting. So when I came to UCLA, I proposed that I apply these markers for radiation to look at different responses. And, we treat every cancer type and we have programs going on in all the different cancer types. But when I first came, I met with Tony Rebus, who's one of the leaders in immune therapy and friendly with our department. He actually did his um, postdoctoral research in our department in radiation oncology. And he's a big leader in checkpoint therapy and developing those treatments. And we were talking, and I was explaining how this class of genetics predicted different patient responses and how our problem in radiation was it would have people that, 15 to 20% of people would have this bad toxicity. And he's like, oh, this is just like PD-1. This happens. People look the same. I give them, and 1 in 4 of them have these really bad autoimmunities, and I have no idea who they are. And it's interesting because before immune therapy, toxicity in medical oncology was, you're giving someone chemo, which is a poison, but they had medicines, right, to help with it. The rare, for your blood counts, for your nausea, they had things to help people through chemotherapy. But immune therapy was a whole new thing where people look the same, but you're poking people's immune systems and they're different. And what's interesting is that's kind of what the toxicity from radiation is actually. We're predicting this late toxicity, which is probably immune based. We set something off, people look the same, but you don't know they have a different immune system, and it builds up over time, these immunities against themselves. So that was our first study with these markers, was in checkpoint therapy, where we predict we can identify people with about a 10-fold increased risk of an autoimmunity, immune-related adverse event. They're called grade 2 or higher from checkpoint therapy. Yes, so that is obviously very broad. I would say there was one interesting lesson I had there. We had this great panel. There were all these companies with checkpoint therapy, and I'm like, it's a no-brainer. Like, one of them should be like, oh, we're the safest one. We're going to test people, and just the people that are highest risk of toxicity, we won't recommend our drug for them. Insurance will make us first in the formulary. It seemed like a great potential partnership with pharma. And we spent years trying to get pharma to work with us. And pharma was like, nope. I mean, what I've learned about pharma is it's about the bottom line. It is about money. And it's not, being creative doesn't pay off because they fire whole departments at pharmaceutical companies all the time. So there's a real fear, I think, of job security. So, I just realized that the timing wasn't right. So we still have that program, and actually it's coming around that patients want to know, doctors want to know, and pharma doesn't have 1,000 salespeople on the street saying toxicity is good. So that program's coming around too. Timing's important for a product. And radiation oncologists are really ready to know why people are radiosensitive, because we've been treating for 120 years and we don't know an answer, but they weren't quite ready for checkpoint. Go ahead.
Thank you. And I'll call on Cindy in a minute, but just we had a session last week with Karen Merritt, who's an advocate for, I think it's DPDY, but it's a pharmacogenomic analysis that predicts a toxic reaction to a chemotherapy.
Oh, good.
And just sort of wonder, how does your tests, how does your thinking fit with more broadly within pharmacogenomics, understanding genetics and then predicting how people will respond to all kinds of therapies?
Yeah, no, it's super interesting. We've really had to think a lot about this because I just had to submit, that's a whole nother conversation, but if anyone's interested, we could talk about it, how you get things reimbursed. And we had to submit a new foundational policy to Medicare because this test doesn't fit into anything. And we looked at pharmacogenomics, and generally they identify like a metabolizing enzyme that someone has a difference in, that then you build up too much, either the medicine won't work because it's not changed in a way that makes it work, or it builds up and it's toxic. What's different about our markers, we're not looking at specific enzymes. We're actually looking at more of a stress response circuit. So it's not tied to like a single enzyme, a single drug. So they're both looking at toxicity, but it's very concrete with pharmacogenomics because they're like, well, it's because they have a mutation in that protein that you need to metabolize this, right? Ours is more about just systemic stress response, but the concept of toxicities is similar.
Okay. Cindy, you have your hand raised.
Yes, hi. Really appreciate your presentation and especially your thoughtfulness in how you formed your company and made decisions who to go with and who not to go with. First of all, I would love to speak with you, offline,
Of
if you
course.
get to share your contact information.
Yes.
I have missed it, but so this is wonderful for folks with prostate cancer, and I guess my question is, if you can identify those that will have a toxicity reaction. It's not clear to me if there's a next step with patients, because I always think about this like, it's really interesting. What are we offering to the patient though? Okay, so what we can offer the patient is they're not going to have a negative reaction. Can we then say for those who will have a negative reaction, but these are the things you can do. It's not what you just can't do. These are the things that you can do. And then the last part of this is there a generalization, an application for this to other kinds of cancer? Is it only, do you have in a sense the model for now trying this with other cancers as well?
That's all great questions, and thank you for bringing those up. So, one of the reasons we started with prostate cancer radiation or prostate cancer in general is there are many options. So, our leading thing is toxicity to an accelerated course of radiation, 5 days. Turns out that the people that are at risk of toxicity to that form of radiation are not 95% are not sensitive to a different form of radiation, which is a slow course. So they inherently still could get radiation, 99% of them, and it just tells you what type to get. They can also, many of them still get surgery. So they have several treatment options, which is great. So that's a great point. Thank you for bringing that up. And the next point is, yes, absolutely. We think, we have numerous programs going on looking at preoperative therapy in sarcoma, looking at toxicity in breast cancer, or looking at response to neoadjuvant therapy in breast cancer and head and neck cancer, which is a very, very toxic radiation treatment. We have panels predicting toxicity, but in parallel also response, because they don't have a lot of great alternatives to radiation. So it's each cancer's nuance, each treatment has its interesting complications to think about. All right, I never want to just give a patient bad news. I mean, that's not helpful, right? I totally agree. And that's been a struggle. Like, someone would be like, oh, we should do a test for that, but it's just bad news. I mean, there's no option for them. So we've been very thoughtful in bringing forward things where there really is a viable alternative for them. So,
Let me
yeah.
just say one more thing. I'm sorry. Then, I think with chemotherapy, there are more companies out there now that are trying to match the right chemotherapy to the right patient or tumor. But I've not heard this, and it just may be me, with regard to radiation. And I'm wondering, are there competitor companies out there? Is anyone else doing this, or is this really your own lane? And that's my last question.
No, but it's a bit of our own lane. Radiation is interesting because it is not FDA regulated, but it's been so competitive to develop the safest way to deliver radiation with the most advanced technology. So radiation is incredibly targeted. It's all about, if we are so focused into this spot. So we use MRI guidance, we use all different types of guidance so we can see exactly what we're treating in very focused fields. The reality is, so we've been doing it for 100 years. We still have people that are just radiosensitive. And I think that we're, finally accepting, it's a very physics driven field, but the biologists are like, people are just radiosensitive. Like you can do all you want, but if you even if you treat a tiny little spot with radiation, they're going to respond negatively. So yeah, so there have been no biomarkers for radiation. I mean, it's been more that it's, sorry, I have a very friendly cat. I'll just try to keep him down. It's been more that, oh, we could just be, we can be smarter than biology, which is impossible, and just focus better. So that's how radiations try to solve it, actually. Great.
Next, Russ has raised his hand. Russ? I mean, Roger. Oh, actually, Roger, then Russ. Let's go ahead.
Unmute. Okay, thanks. I have 3 really quick questions.
Sure.
Joanne, you might not remember me. I talked to you about a year ago.
I do.
Oh, okay.
I'm like, I remember your name, but I hadn't seen you. We didn't, oh no, we did have a Zoom, actually.
Yeah, so just for everybody else, I had an experimental neoantigen peptide vaccine about a year
Yes.
ago. My doctor pushed me really hard to get a checkpoint inhibitor along with it.
Yes.
I mean, really hard. He said, this does not work without this checkpoint inhibitor. And I got your test, and it turns out I'm one of those people that would have had a grade 4 adverse reaction. And I took it to the doctor, and he never mentioned it again.
Wow.
Since then, I've talked to several other oncologists, and it feels like nobody knows about this. And
Yeah.
recently, I was up at UCSF,
Yeah.
and they had an oncology group, and I talked to somebody there who they gave a presentation on checkpoints, and I said, well, what about adverse reactions? And they were like, well, that hardly ever happens, and even if it does, we can treat it. And I said, why don't you test for it? She says, there's no such thing. And I connected that with your website, and she emailed me back, and she said, holy cow, this is real, you know?
I know. It's thank you, Roger, for bringing that up. That was part of the battle. So we had this great panel and no one wanted to hear it. You know, it's like, I was like, we can't pursue this right now. We're going to lose. I mean, when we started with this ovarian cancer risk prediction test, that was my experience. Our timing was terrible. Myriad bracket was going off patent. There was all this animosity, and they had 300 salespeople like saying bad things. I'm like, that will be a losing fight right now. The time will come. We'll continue to publish on it. We'll continue. We have another independent validation that we're presenting at ASCO, actually. The time will come and it will continue to mature. The numbers will increase, and we're just about to start an early access EAP, early access provider testing at UCLA for this. So I'm letting it do the groundswell. I mean, that's what we did with the prostate test too. But I love if a patient knows about it and asks for it, I want to make sure they can get it. So it's a developed test in the lab. They can get it through Mirakind, you know, which is basically how we got that to you. So thank you for bringing it up. That's part of the challenge and the battle.
I mean, if this were a drug being promoted by big pharma, I'd be seeing commercials on TV every night.
I know. But the problem is you have someone that's like, no. You know, pharma, when we started our first paper about that toxicity, it took 2 years to publish. That's like a record, because there were reviewers that like, no, pharma's telling me toxicity is good. Toxicity means you're going to respond, which is absolutely not true. And they literally, we couldn't even get the paper published. It took 2 years. I'm like, this is so political right now. It's so politically charged. So, but yeah, thank you. Yeah.
So quickly, I asked you before, and at that time, the answer was maybe later. It was, do you have a solution for anti-PD-L1 antibodies?
No, yeah, we haven't tested that yet. So do you mean the next level of predictors for those?
Yes.
Yeah.
Right.
Yeah, no, we haven't done more. Some of it's getting access to those trials. I mean, radiation oncologist, it's easy. All we need is patient cheek swabs. But when pharma owns it, they won't, it's very hard to get samples from them, right? To predict anything they don't want. They like to control the data. So if the newest drugs, it's almost impossible to get access to because they control them. So they won't let us do it.
Yeah, and then finally, quickly, you know, I've been hearing more about radiation toxicity, and I'm just wondering, and I guess that is pretty much one of the things you can test for. Can you speak a little bit more to that? Because, you know, I don't know, I don't hear a lot about it, and I think maybe I should.
You know, I would, compared to how we used to, everyone's scared of radiation. And there's like the article front page of New York Times, it was like, there was a mistake in a treatment and someone had terrible side effects. In general, radiation these days, it's so high tech and it's generally safe for most people. But, you know, as I said, there's a subset of people that will develop late fibrosis. As medical therapy is getting better and people are cured from their cancer, the aspect of long-lasting side effects is really important, and it's been largely ignored. We've been so focused on like, what can we do to cure people? Pharma leads that this drug will cure you, this drug will cure you. There's been very little conversation about people that are cured and forever are affected by their treatment. And I think immune therapy is really also brought it to the front, because these autoimmunities, you can have become diabetic, you know, you can go blind, you can have liver disease, you know, all types of things, cardiomyopathy from these autoimmunities that the medical oncologists will say, we cured them, but we've killed them, basically. And so I think that's helped bring it to the forefront. Radiation, as I said, in general, severe toxicity is incredibly rare, like less than 3% of people will have a grade 3 toxicity that is long lasting, but it really matters for them. And, you know, our test for prostate cancer can predict about a 4 to 6-fold increased risk of that. So that's meaningful versus being very low risk for it. And right now, if you tell someone, oh, you're at about a 3% risk of a really bad toxicity, it sounds really low, but they're the people that are really at risk of that. It would be fantastic for them to know and do something else. So I hope that's the future. The problem is pharma's never going to be on board with toxicity predictions because they will lose patients if someone's going to have toxicity. I mean, I remember a conversation I had with like the head of science at BMS about our checkpoint inhibitor toxicity. And he literally said, wait a minute, if you tell someone they're going to have toxicity to my drug, they're not going to get it. And I'm like, yeah, right? Like, obviously. And he's like, yeah, I don't think we want that. So that's a struggle. And so I think it's an incredibly untapped, incredibly important area. So,
Thank you.
sure.
Russ, you had a number of questions in the chat. Could you choose whichever ones you want to bring forward?
Okay. First, I might have missed, I was looking at some stuff, and so I might have missed this. Is this the Prostate Ultra test?
Yes, so that's the first test that's
Okay.
out there.
Okay. How does it compare to the MIR Sentinel?
MIR Sentinel. I'm not sure what that one is. Is that, can you tell me what that test is?
I just know it's a microRNA test too, and it's FDA approved, and it's it's now say like I assume similar to yours, but I don't know what the overlap is.
Oh,
Just
I don't
curious if
know,
you're
actually.
there with me.
What's a path to confidence? Is a standalone? Oh, so that's diagnosing you with prostate cancer. I mean, there's a lot of efforts. There's been a lot of work in prostate cancer to, one, figure out which tests need, which patients actually need treatment, and actually to diagnose it. You know, it was all on the PSA before, and PSA is not specific enough, right? So, yeah, so there's a lot of technology looking at that. So that's actually looking at, I think, microRNAs in urine to see if you have prostate cancer.
Okay, so it's a prediction test.
Yes,
Okay.
but it is actually using microRNAs, which is kind of the same concept.
Okay, and I definitely don't need prediction. I have it. Uh, can it possibly give you insight into meta development?
For the risk of metastatic disease?
Yeah, the risk of metastatic, give you like a heads up, crudely. You know,
Yeah,
there's
well,
lots
so
of
the
things that
test,
can.
the Prostax Ultra test doesn't, it's really looking at your response to a radiation treatment. We do have a lot of work at looking at, you know, tumor aggressiveness. You know, a lot of that's done by looking at the tumor. In our platform, we're mainly looking at the patient. So we're looking at the patient's DNA versus the tumor DNA. But there's a lot of work, Decipher, a lot of things, a lot of tests in prostate cancer looking at how aggressive is this tumor and what are the chances that it would spread.
Okay. Does it happen to look at AR upregulations, mutations, activity?
So our test doesn't again, but there's a lot of interesting work doing that, right? Looking, because a lot
Yeah.
of that, looking at the androgen receptor and responsiveness to some of those very good targeted drugs. Yeah, nope. So since we're not looking at tumor, we're not looking at that. We're looking really more at the patient and their response to things.
Okay. Yeah, okay, so I assume with the radiosensitization, could you, like, say, for example, before I did a SBRT,
Yes.
gotta take get a baseline and then take olaparib or some PARP inhibitor or whatever I thought would radiosensitize me or what, and then redo the test.
Oh, interesting. We are really looking at the test isn't actually looking if in general prostate cancer, when we treat it with definitive radiation, it works pretty well. The local, it's pretty, people can fail after radiation, but the control rate is pretty good. So we're not actually looking at sensitivity to radiation in the tumor versus sensitivity of the patient where they will have side effects from the radiation.
Hmm. Okay,
These are all good questions about products that there is work in those areas.
Okay. Okay. Would it be possible to, I guess so. Okay, so it's not the tumor, so you had to extrapolate to the microenvironment, whatever the case. So
Yeah.
oh, I just had a comment about toxicity. Have you tried with the pharmaceutical companies to have an opposite approach and say, look, you know what, we could screen people out, but we could also screen people in. Like, let's say Russ Hollier is, for example, he's really afraid of toxicity. I am. So
I
if I can
know.
tell him, hey, look, you're on the low end of the toxicity rating, I'd be like, okay, cool, I can do this. This is an option.
I know. I wish if they would talk to us. We may try again. We're actually doing some work with lutetium, which is, the systemic therapy, like a radiation labeled
You ligand.
antibody, basically. Yeah, in prostate cancer, we are looking at some of our microRNA signatures, looking at who will respond. There's just not very many side effects with that, actually, but some blood count side effects.
Okay. All right. Well, thank you.
Of course. Yeah, my pleasure.
We've had Oliver Sartor speak, and he's a good friend. If that would be useful, he's sort of the lead PI on all of the radioligand therapies. So
Oh yeah.
we can make that connection. Rick Davis is up next.
Well, I got a lot of things to, I'll try and be short. I got a bunch of things to talk to you about. However, one question just came to mind, which comes up all the time, and you may have the answer to it or may have researched it. Have you found or have you been able to track whether people with germline BRCA are more sensitive to radiation or
It's
not?
such a good question. As a breast cancer researcher, we've done, there's been big studies trying to look at that. And this is what I would say. I think that an important thing about radiation, there are really two types of side effects. There's short-term side effects you can experience during treatment, and then there's long-term side effects that actually don't even develop until 9 months. I do think people with BRCA mutations, ATM mutations, they have worse short-term side effects. The studies that looked at it looked at like overall side effects, and I've treated some ATM patients with breast,
Right.
they're radiosensitive. I mean, that's how you diagnose ATM mutations, and BRCA is involved in the radiation response too. So I do think they have worse short-term side effects. They do not have or necessarily have long-term side effects. And we actually have genetic panels, microRNA panels of short-term side effects. They're all about DNA repair, which is what ATM and BRCA do. That's not what our late toxicity panels are about. They're more immune-based because that's more fibrosis. So I think you can quote me. Yes, I think they have a higher risk of short-term side effects.
Thank you. It's an ongoing debate, especially when we get the guys. We don't just do prostate work with prostate, but we work with a lot of prostate. And when we get these guys who come in who are like Gleason 3+3s and 3+4s, and they're deciding what treatment to get, and they need to get treated because they carry BRCA, and should they do surgery or should they do radiation? And there's always a discussion of, well, if you did radiation, you'd be more sensitive.
You're a
So
little more sensitive. The good news is actually, we usually can get people through the short-term side effects and they go away. So, you know, they go on Flomax or something just to help through the acute side effects.
right.
And with breast too, you know, if they'll have worse skin peeling, but it heals and actually they look fine. It's really the long-term ones that are.
Now, I have the t-shirt, so I got a lot of radiation, and I do have the t-shirt, and I do have long-term side effects. But as I always say, I'd rather be this side of the grass than the other side of the
Yeah.
grass, right?
We'd like
So,
to do
and
it both
I
ways.
would have been the other side of the grass. So,
Love to
you
get
know,
you there without side effects. I think that's the dream
yeah,
future.
yeah. Well, my doc was Matt Roach, and
Oh,
I've
Mac
had
uses
discuss.
I love Mac. He uses the test all the time. Prostate.
I love Mac too, and
He's so funny. He's such a
I love
character.
Mac. He's yeah. Now I'm going through a three-way conversation here. You, me, and the lead moderator of our low intermediate group, prostate cancer group, who lives in LA,
Yeah.
Dr. Mark Perlow, who's a good friend of Kishan's,
Oh,
and he works with Kishan all the time. And he's
yes.
sending me questions to ask you. And I said, no, I'm just going to give you Dr. Joanne's,
Yeah.
uh,
That's
and he can ask you. He'll be in touch with you.
no problem.
I just want to tell you, we have been using your test for donkey's years. I mean, we've been using both tests, Prostox and Immune.
Oh, good.
We don't see a lot of immune checkpoint because, as you know, it's a cold tumor, and unless somebody is Lynch or something like that, they're not really a candidate. But we have had people who have been candidates for pembrolizumab or nivolumab who have wanted to test whether they're going to be sensitive. And so we love you. We love your test when we need
I'm
it.
so happy.
Yeah. And the people in our low intermediate Prostox, they meet twice a month. Prostox gets talked about at least 50% of the time, at least 50% of the time.
Oh, well, I'm happy to
And
get some.
I would love to invite you at some point. Maybe we can do a
Yes.
session. And
Awesome. Yep.
also, I would like to make available to you our guys for your own research, so you can ask them
Fantastic.
questions. You probably know Pamela Munster at UCSF she's the BRCA lady.
Oh,
She
okay.
she's the lady who actually has BRCA, and she runs the BRCA clinic,
Okay,
and
good.
she's got a prostate cancer product she's working on. And she's
Sure.
come in because she wants to talk to the guys directly and ask them questions. If
Awesome.
you'd like to do that, we would love to have you
Awesome. I
and
love it.
work with you and give you the opportunity to ask these men what's going through their mind when
Fantastic.
they want to take.
I love to
So,
talk to patients. I mean, that's why we have Mirrorkind, and I have people join a registry because I'm like, I'm going to follow up with you. And in our
yeah.
breast, we have a lot of breast cancer patients on it, and I did a big study, and I'd send out communication, and I would literally have 1,000 emails in my inbox the next day, and I loved it. And I'd answer every single one. That's kind of what I did during business school.
Okay, I won't take more time, but I do need you to put up that your contact information screen again, and we'll
Okay.
be in touch.
Absolutely. All right. Oops, I got to share again. Did I stop sharing? Sorry. Share there. I didn't mean to take that down. Yes, everyone, please fill. Oh, I did that so I could see everyone, but I still can see you.
Okay.
Yeah.
Okay. Could you make it a little bigger for
Yeah.
my old eyes,
Oh, yeah,
please? Yeah,
sure.
just on the presentation.
Or maybe
you
just put it in the chat.
There we go. How's that?
Okay, I can work off of that. And thank you very much, and we'll be in touch. It's lovely to meet you.
You too. Thanks so much. I hate when it does that.
Okay, next up was Richard Anders.
Yeah, this is really interesting and great work, and I really am impressed and admiring of the work you're doing. I just have a few questions because I'm always curious about sort of the science and some of the things like that. First of all, are you a CLIA test, or are you FDA test, or both, or?
We're CLIA CAP certified, and actually we're having our New York State inspection. There's this whole big debate about the FDA, and this whole rule just changed that it's
Yeah.
no longer regulating these tests, but we're a CAP certified lab. That's kind of like the step up, and then New York State is coming in 2 weeks.
So I put a question in the chat, but it sounds like you've kind of answered it, which is the toxicities that you're looking for in radiation are things like fibrosis and other things like that. So probably it's not like those are not things where if the cancer happens to have the same germline mutation, you care that the cancer will develop fibrosis. So it's not really relevant to measure susceptibility or anything. You're measuring longer term effects that a patient might use to say, if there's a little bit of off-target radiation, steer clear because you could get these adhesions and they could be really bad unless you can be very focused in the radiation, perhaps.
Yeah, so I would say, I expect we do have studies looking at both response as well as toxicity. They don't seem to be the same pathways. I thought maybe they might be, but they're not. And for prostate, what's interesting about prostate, what we're predicting are your late urinary side effects. Because we treat the whole prostate with radiation, I mean, we tend to treat the whole gland, and the ureter runs right through it. So if you get fibrosis in the prostate, you're symptomatic. I mean, you just are up every half an hour, you have to go to bed. It hurts to urinate, you're urinating half an hour, or it could be worse. So that's the fibrosis of that gland. We, of course, look at those signatures. Every cancer has different toxicities caused by different things. So in breast cancer, you can have fibrosis that would be misshapen, changes in the look of the breast. And, if someone has a reconstruction, it can be very problematic. Sarcoma is something we study. You can get fibrosis of the joint. Head and neck fibrosis really causes like permanent swallowing issues. So it kind of depends where we're treating those tissues, what the side effects are.
So it's really, the fibrosis seems to be the major thing you're predicting, which
I
is,
think so. For late toxicity, we think that's generally it's a fibrotic response.
so that, it's like telling someone they'll get keloids or something on, whatever.
Yeah,
That's really interesting and very, very helpful. On the immune checkpoint stuff, that one's really interesting because I would imagine that a lot of that has to do with the particular immune repertoire of a particular patient. But you seem to be possibly saying, unless you're measuring the immune repertoire, that there are certain people who tend to develop autoantibodies in general. If that's true, is that test good for autoimmune diseases in general, or is it specific to autoimmune checkpoint inhibitors?
Such a good question. And actually, I'll give you an example that I think kind of will explain how the biomarkers work a little, and it is in autoimmunity. So one of the first examples of the type of mutations we study was in Crohn's disease. And what they found were people, autoimmunities, they run in families, but not everyone gets it. So it's never clear exactly what it is, who will get it and who won't. But it's always been thought probably something sets them off. And we think, for radiation toxicity, for example, people walk around with these differences. They're fine, but if you radiate them, you set them off. Or with immune therapy, they might have different immune systems and they're fine. But if you poke their immune system with this pathway affecting, PD-1, CTLA-4, you set off a bad cascade. So this example in Crohn's disease, they found just a single difference in the parts of the DNA we study. And it was in a calcium channel, basically in the gut lining. And if someone was exposed to a specific strain of E. coli, a bacteria, it would set that off, and it basically got locked on. Basically, it got in a vicious cycle and that channel stayed open and they developed Crohn's disease. So it was all about the exposure tipping something off. And so I kind of think of it that way. And I do think that even in the radiation patients, some of these markers are probably important in autoimmunities. A lot of the panels we study are genes that are known to be important in autoimmunity, but we are very bad at predicting who's going to get it, how to treat it. and figure out what the triggers are. So that's a whole nother area where I do think these markers are very relevant, actually.
Well, I mean, there's all these tantalizing clues, like, you know, different HLA types get certain preferential autoimmunities, and then, you
Yes.
know, the MS and EBV is sort of a proven correlation more recently. So do
Yep.
you look at stuff like that? Are you looking at HLA type as part of your test?
We don't look at HLA type, although there is definitely some implication of HLA type in immune-related adverse events, but there hasn't been a great development of that. There are a few other germline biomarkers. Our real goal with the germline biomarker approach is being able to test somebody before they get any therapy, because once you've gotten the therapy, it's actually too late. And they may say, oh, if you get immune-related adverse events, we can fix it. You kind of can't sometimes. I mean, and radiation toxicity too. Once you start it, you started down a bad path. So even these autoimmunities, they do check people, and there's a lot of debate about giving immune therapy to people with autoimmunity, and they can measure some of those things in the blood before, but there are a lot of people that don't have those yet. Right, but they might have the genetics that will get them there, and we're trying to identify those people, so they don't know before treatment that they're going to go down a bad path.
And last question, sorry. So you have studies and whatnot that, I mean, there's a lot of, and it's not a well-kept secret anymore. It's not even a secret, but there are a lot of people who get some level of autoimmunity immune problem with checkpoint inhibitors. And it's just a question of how well you can control it. But do you have any data? Of course you do, on how much you enrich the patient population of people who are going to have adverse events, and, you know, maybe if there are papers, I'd be interested in seeing one. You don't need to answer it now. I know there's another question at least.
Yeah, no, we predict about a 10-fold risk. So that's like,
wow.
Yeah, I know.
Wow.
The differences we see are like 8-fold, 10-fold. They're not 10%, right? They're really, really identified people. Like for a prostate toxicity, we identify people, separate people from the average of 20% to 4% to 80%. Right. So we really differentiate, and the same with autoimmunities. That's meaningful information for patients, like a 10% increase or 10. I don't think that's very helpful. So we're really happy that there are big differences like that.
Honestly, if you were telling me that you do a 10-fold increase, I would say you'd be 180% or something. I mean, a 10-fold increase is enormous in a group that has a lot of that. Wow. Well, that's great. Thank you very much.
Of course.
Elaine Watkins is up next.
Hi, so I had my question is tied to the previous. So for a patient who has any autoimmune tendency, meaning
Yes.
they've either expressed antibodies in the past or maybe currently don't, those patients are contraindicated for almost all clinical trials.
Right. Yeah.
And I'm in that category. I had no autoimmune disease until my first chemo treatments.
Oh.
That triggered it. And when my symptoms have been triggered, it's poking. Proton radiation did it.
Huh.
So, but the problem is I can't get into any trials because of the autoimmune condition.
Yeah.
Now, the flip side of your argument that Big Pharma wants everybody and anybody in their trials is not quite true. I worked in Big Pharma.
Yeah, I'm sure it's true. They don't want
So
people that will have bad side effects if they know, or if it's obvious. Yeah, I mean, I wish that they would let us do that. I mean, it's such a no-brainer to me. Like, take out the
Well-
bad people, you'll be approved. There's so many failed.
Oh, correct. It's called cherry picking, by the way.
Yeah,
So
yeah,
Patrick Shung or whatever his name is, he's
Yeah.
notorious for that. His drugs are remarkable. But you know what? He doesn't touch a patient like me,
yeah,
right? But I need his treatment. That's
I
the
know.
treatment I need. So
right.
my question to you is this. If I go to Mayo Clinic, my clinician at Mayo, and say, hey, I want this test, is it ready for prime time for a patient like me?
Yeah, I mean, so the checkpoint inhibitor therapy?
Yes, any immunotherapy, vax or otherwise.
Yeah, I mean, so the studies that we have are in checkpoint therapy, and I will tell you, or PD-1, we actually have a CTLA-4, but no one gets that alone anymore. And we just did, so our first study in that was in very advanced melanoma patients. And I was worried because I figured they were pretty genetically pressured because it was a phase 1 trial and they had been through everything. So we actually did it on a much broader population, about 250 patients who were getting it as first-line therapy and have, expanded our panel. We won't always catch I mean, someday maybe we will, but as we expand our cohorts, there'll be people that have rarer combinations, which are underrepresented in the people we've studied, right? So we won't catch everybody. But I would say usually if we say you'll have toxicity, you will. I mean, the chances are really high. It's not 100% you won't if, we say you don't. So I would say our prostate one's further along mainly because it's been easy to get these validation cohorts. Our immune therapy is close to coming next in line. We're planning to open this is an early access program at UCLA because there really is a lot of interest in trying to predict it and figuring out what you do and what patient populations would benefit from knowing, right? Because some people really need to get the checkpoint inhibitor. So, we'll have to work with the clinicians on that, but always happy to.
So it is prime time. I could get the test.
Yeah,
At least it would tell me something, right?
Yeah, I'm happy to, we can connect and I'll
Right.
figure out what we can do.
All right, thank you. Thank
Sure.
you.
Joanne, we're at the top of the hour.
Yeah.
How are you in terms of running over?
It's okay. I can do it.
Okay, so let's wrap up the formal portion here. If you would just, your parting words of wisdom, any final thoughts? I'll stop the recording and then we can have a little informal discussion.
Sure. Okay. My takeaways are I love talking and connecting. Thank you so much for the invitation. It's been wonderful and for the great questions from everybody. I think there is a lot to still do. And, we need patient and advocacy group and everyone's involvement to move things forward together.